The Jeff Gabriel Connection.
Hey, George, how are you today? Welcome to the Jeff Gabriel Connection. How are you?
I'm great. It's great to be here.
I just want to give the audience and people that will eventually be listening to this just a little background about who you are, what you do, not known because you're already so well known. You worked to initiate the Human Genome Project and the Personal Genome Project in 2005, which provides the only world open source genome map of the DNA, which really opened it to many, many people to see what mapping of the genome was all about. Your early work using X-ray pathology techniques and to study the three-dimensional structure of transfer RNA, which communicates with cells, was groundbreaking and occurred early on in your career or your early studies at Duke. You're considered the father of synthetic biology. And I want to come back and discuss this a little bit later on and some of the maybe social, ethical implications of that. You're a geneticist, a molecular engineer and chemist, and you're head of genetics at Harvard Medical School, head of the genetics lab with more than 100 on staff, and you are the author of Regenecist, How Synthetic Biology Will Reinvent Nature in Our Cells. And I definitely want to come back because it's so interesting. Given that the average podcast listener is around 24, a lot of podcasts where people are extremely educational. So George, it's not just information. They want to know a little bit about you because you've had such a migration and such a path to an incredible career. But my first question for you is, how does George Church spend his day?
Well, quite differently during the pandemic, as is true for many people. It's a mixture of programming, computer programming, and talking to lab bench scientists that are working on a variety of biotech and synthetic biology and clinically oriented technology development. That's kind of the day to day. If it's not a pandemic, I used to travel quite a bit. During the pandemic, I kind of doubled down on programming and writing.
So you're also a computer scientist as well. You're a programmer, not just a biologist or a geneticist.
Yeah, I've been doing some kind of programming since I was 13. Which was I'm now 66, so it's not a full-time thing, but it's in a way it colors the way I look at technology development in general.
Right. There's some similarities between designing and biology and chemistry and how you look at the genome as well. And we'll get to that a little later. And I'm going to start off with some simple questions because we want to know a little bit about you, not just the work. So you're a young boy or you're adolescent, teenager. When do you say to yourself, when do you get this bug? Because usually when people get as refined as you are in your profession, it starts early. It's not a later in life decision. It's something that you figure out early on. And when did that light switch go off from you? How did you know?
Quite early. Not because it was the family business. I didn't actually know any scientists or engineers, and it wasn't really taught in my school until fairly late. But easily in 8, 9, 10, I was exploring kind of the wild part of my environment, the canals, the mud flats. I lived on the water almost my whole life until I went away to Massachusetts from Florida. But I was also inspired by my third father's avocation as a physician. So he would do house calls and things like that, and I thought that was interesting.
So while other kids are outside with a baseball and the mitt, you're outside behind looking at the streams and studying potentially bacteria or different amoebas floating by, that kind of thing. You had the bug early for sure.
That's right, and it got in a big injection when I went to the New York World's Fair in 1964, where I saw the expectations were high, and all the countries of the world had really gone all out to create their vision of the future, and I just totally bought into it. I was, it was just the right age where you're easily impressionable, but not quite skeptical yet, and so I saw robots, and I thought, oh, that's, let's program computers. So.
Yeah, I was mesmerized by robots as well too, and maybe this, this a lot better than I, but I'm just thinking out loud. Maybe some of this, there are genes turning different things off on our brain, allows someone who's younger as opposed to older to be able to see like you with the clarity that you did, that of your interest in following this. But the next question that I wanted to ask is, can you give the audience, as I said, some of the audience is probably young, just a 30-second primer of DNA, and do you look at DNA the same way you looked at it 10, 15 years ago, in contrast to what you know now and what studies are showing you and what you've learned? Do you see it differently now, George?
Everything changes a little bit. I'm not that easily surprised, but I think that maturity in science. I first I saw science kind of the way you would you get it from popular biographies of Madame Curie or Edison, Einstein, and that isn't quite as realistic as and then there's science fiction, and then there's kind of like what you would like to do to make something explosive or smelly, or the kind of chemistry ideas that a 9-year-old might have, and that evolves over time into what is the reality, and I think a much more exciting reality, where you have amazing ability to participate in, say, an exponential economy, where fairly simple ideas that you have can take root, and you can see them grow into things that affect people's lives. That's a very different vision of DNA than one might have when you read Watson's The Double Helix or something, which is much more like a sporting event or a simple molecule. It's a much more complicated ecosystem, is the way I look at it today.
When you first started studying DNA, were you amazed or intrigued by the symmetry of it visually, how it looks? Because it looks designed.
Both DNA and transfer RNA are beautiful molecules. I mean, they really are. DNA is very much more symmetric than transfer RNA, but transfer RNA essentially takes that symmetry and twists it around and embellishes it in such a way that it looks like it does more. The problem with DNA is you look at it, it's so symmetric and so straight. You sort of say, how can that do anything? It's like a wood screw or a plank of wood. But what is sort of beguiling in its simplicity is just any string of A, C, G, and T has a lot of information in it if it's long enough. The same as a string of zeros and ones has a lot of information. That's how we're recording this conversation, is a bunch of zeros and ones. And so that simplicity makes it easy to get into, but it also allows you to sequester and use a lot of information.
But again, when I look at it, and again, I don't get to look at it under a microscope, you were amazed by the structure and how it looks. I mean, did that kind of blow you away a little bit of what it actually structurally looked like? I mean, it looked like someone sat down as an engineer and put it together. I mean, and it's evolved over millions and millions of years. I mean, to me, it just seemed interesting.
Yeah.
Or maybe it's just a little bit too way out there, that question.
No. I think all of that was true and more. I mean, there's just many, it's like a poem or a scene, a beautiful scene. The more you look at it, the more you see, and the more different ways you can see it. So evolution is one way, symmetry is another. I had the great pleasure of starting looking at it as a crystallographer, which was the whole field of crystallography is very much focused on symmetry and understanding things at sort of ultimate levels of resolution, where you can actually see electron clouds and you can see the bond, the separation of atoms at and that's just somehow being able to look at the flower as a field of flowers, but also at the atomic level gives you a whole range of appreciation.
Yeah, for sure. Given that genes are the building blocks and they are the menu, the structure for our growth or development, and you can correct me if I'm wrong, because again, I'm speaking as a layman, how I'm interpreting your work and how I'm reading this. The majority of your time in the lab, would you say, is dedicated now to gene therapy and the ability to take the bad out of the genes and recreate the good to rebuild into something healthy? And I believe the CRISPR technology that was developed allowed you guys further access into developing these techniques. Could you elaborate a little bit on that, if you don't mind?
Gene therapy is one of the avenues we're pursuing. It's not necessarily the main one. We've innovated with gene therapy and making better ways of delivering the therapy, as well as different targets for it. And it's not just fixing broken inherited genes. It can also be used for vaccines. So in a certain sense, the Moderna and Pfizer vaccines are a kind of gene therapy where RNA is being delivered. But it can also act. You can get at some of the same features fixing genetic diseases by genetic counseling, which is more based on reading the DNA and taking personal actions on it, rather than gene therapy, which is relatively expensive so far. So we're constantly looking for things where we can get access to everybody on the planet, so make it equitably priced, so that everybody can benefit.
But the ultimate goal is to be able to go in and fix many diseases, to make people healthier,
Right.
to maybe even one day with an ethical issue is to enhance. I know I'm not here to judge that or anything, but that could happen one day. You could use gene therapy to enhance, correct?
Well, actually, the almost all technology is enhancing. Cars, jets, fire, wheel, you name it.
Right.
And the vaccines are a kind of gene therapy. You're changing the genetics of your immune system via the genetics of a disabled version of an infectious agent. So that goes back to the dawn of vaccines to smallpox, and that's an enhancement in the sense that if our ancient ancestors could come forward in time and look at how none of us get all the diseases that were wiping them out, they would say these are superhuman. But we're not superhuman compared to each other, just compared to our ancestors. And we'll continue to do that, continue to make enhancing technologies. The important thing is that they're safe, effective, and equitably distributed to everyone.
Yeah, well, I'm sure 1,000 years from now, the futurists or the people in the future will look at us and say that we were ancient. I wasn't going to talk about the vaccines that much today, but I know you've been working with it. And a lot of people are scared of the vaccines. A lot of people are taking the vaccines, George, but a lot of people don't. Again, I wasn't even going to talk about this, but now that you brought it up, it's really interesting. A lot of people are nervous about the vaccines. Could you explain how the RNA vaccines work and why they're safe, or what you think about them? It would really be helpful to the audience.
Well, first of all, I think it's good to be nervous about new technologies. I think it's good to be nervous about cars. Cars kill 1 million people a year. But it's really all about the benefits versus the risks, and the benefits of not being one of the 3 million people that are dying of COVID-19 and a variety of other diseases like smallpox that are now extinct because of vaccines. So no technology is without risk, but here unquestionably the benefits outweigh the risks. Just because you know an anecdote of someone who got a particular disease, measles, smallpox, you name it, and managed to survive, doesn't mean that you will. And so it's like in Dirty Harry, do you feel lucky, punk? And I generally don't feel lucky. You take the unquestionably what route to wellness, which is vaccines.
Right. Dirty Harry was a major fan of him. I actually met him one time, the 1-shot versus 6-shot guy. So hopefully the 1 shot that everybody's getting is the positive, the good one, and the healthy thing. What do you think will be the outcome of COVID given the vaccinations around the world? I mean, hopefully positive. And I mean, you're a very smart individual, and your take on it would be appreciated for me today and those listening. I'd be interested to hear what you think.
Well, I think it's similar to seatbelts and smoking. It is you sort of, a lot of people feel lucky, and they but for the good of your family, think about your family. You want to protect them, and so you want to take the best shot you can. I think that this very unfortunate year of COVID is going to have very positive consequences of various sorts. I mean, I think we've gotten alternatives to travel, like the kind of video telecommunications that is so much better now. We're taking science, I think, particularly biotech and medicine, much more seriously. And even if not everybody is impacted, or not everybody takes the lessons home, enough people are that I think that we're going to have positive economics and positive health benefits from the number of people that are now taking this kind of preventative medicine seriously. Preventative medicine is probably a lot less expensive than trying to deal with emergency medicine and reactive medicine.
Well, that's really interesting. And I definitely run across a lot more people that are interested in taking the vaccine than are afraid of the vaccine. And it's good to hear that you think it's a good thing, and you think it's potentially reasonably safe and a reasonable risk to take. And everybody should follow your lead on that. I wanted to switch gears a little bit, and I want to talk about something that you could explain. I know what a virus is, but what can you explain in your terms what a virus is and how it interacts with the body, in the sense? I mean, I know you can deliver good things with viruses, and then viruses can get into a cell and screw up the communication, and then the replication's off, and then the cell gets unhealthy. But is it like a ghost in the machine, George?
Yeah. So viruses are fairly ubiquitous particles. They're not capable of division without help from a cell. So they have to invade a cell in order to make copies of themselves. They're somewhere in between molecules and cells, but they do replicate in the right conditions, and they sometimes they do it without impacting the host. In fact, it's to their advantage that their host proliferates to provide more hosts for them. But every now and then there'll be a virus that's out of sync with its host. It'll either be recently come in from another organism, like from an animal to a human, and they'll be well suited for the animal, but they might be either too lethal for the human, or can't replicate in the human. But anyway, they're just not well suited, and we need to watch out for that. There's increasing amount of humans disturbing the natural environment, and hence inviting zoonotic diseases from animals to get into us where we're not well adapted. Viruses are this replicating entity to which your immune system can sometimes fight, but usually it needs a little bit of help. It needs a chance to learn. The immune system learns its enemy. It can learn its enemy on a vaccine rather than learning it, on the, in an emergency situation. If it's learning its immunity while you're dying, that's a little too late.
So it always needs a host. I mean, is it jumping around by itself? I mean, it has to be in a body, it has to be in an animal, and its origins are inside. It actually originates itself inside of a body or an entity.
It does, and it can spread by a variety of mechanisms. It can spread through the breathing through basically through the lungs. It can by contact. Different viruses have different means. It can spread through your food or water if it can survive your digestive system. Those are the main methods, and some of them can be eliminated by good public health. Drinking filtered chlorinated water rather than what we used to do, which was to drink from the same river that we would defecate in, which is not smart. But we didn't know much science back then.
George, I try not to do that.
Yeah, exactly. So you've benefited from hundreds of years of science.
Yeah,
It seems
exactly.
obvious to us now. A lot of things that were once very scientific are now common sense.
Yeah. Well, back in the 60s, we'd like to throw litter on the ground, and we didn't care. So now we pick up and put it in a can. So we're always learning. Were you a fan of the Matrix movies at all? Are you a sci-fi guy?
Yeah. Definitely enjoyed the dodging the bullets in slow motion. That was kind of cool. Yeah.
So I got to ask you, I got to just step out of the serious talk for a second and just talk about the Matrix. So when Smith, is talking to Neo and basically describes human beings as viruses, I'm not insinuating that human beings are viruses, but do you ever study, when you look in the genome, when you look at genes, when you look at viruses? It's an interesting question. Do you ever see behaviors that, sort of correlate to human behaviors we're more complex, it's a little bit simpler, but do you ever see any patterns that say, that's interesting to me?
Well, we all evolve, and there's not only evolution of molecules and organisms, there's evolution in a cultural sense. You can see certain ideas proliferate and get selected. So replication, mutation, and selection are the key components of any kind of evolution, whether it's cultural or biological. So I think that's the sort of thing that Agent Smith might have been referring to. It's a poetic, metaphoric license. I think it applied. I think too often people they only think of mutating DNA. They don't think of the evolution of our technology and culture. There's a lot of trial and error in all of these things.
Right. There was a lot of negativity, but there's also a lot of positivity that's gone on our culture. And, the news and our culture likes to spew the negative, but as you mentioned, there's a tremendous amount of positive things going on. And I think we need to focus on that for sure. I wanted to ask you a little bit about your theories and your thoughts on aging. I know you've talked about this a lot. And I believe, and I've researched a little bit how you've spoken about this and how you've researched this, but I believe that you were one of the first people that said that you believe that humans can live a few hundred years.
Yeah, so we do research on this, and we're moving into clinical trials, both for veterinary purposes and for human clinical practice. Our emphasis is more on aging reversal than it is on longevity. And part of the reason for that is that the amount of variation in longevity between you and me makes it hard to prove that I've got a longevity drug in less than 30 years. And a 30-year clinical trial is expensive and hard to get people to sign up for it. But aging reversal, a huge fraction of human morbidity and mortality is due to age-related problems. And many of the symptoms of age-related health issues you can see reversal in days or weeks rather than decades. And so now it turns out that if you can do aging reversal, then you can also get longevity. But the point is you have an immediate FDA approvable benefit that you can see in a short period of time, and then time will tell whether it also has longevity effects. I don't know what the upper limit is. I don't know of any law of physics that would prevent us from living a long time, other than the end of the universe where it all cools down. But that's so far off that we don't need to worry about that just right at the moment. So I think aging reversal is where we want to be.
I'm the kid in the back of the classroom right now, and I'm waving my hand because it's really interesting what you said about aging reversal. So when you say aging reversal, are you stopping the aging, or all of a sudden, George Church is 25, he looks 25 years old again, or you can bring him back 5 or 10 years? Can you go back like that physically?
So you can take subsystems at least and go backwards. There's at least 9 different pathways of aging that occur in your body that are understand quite well, especially in animals. And you can take, and some of those are associated with multiple failures that occur as you get older. It's kind of like a planned obsolescence. You've reproduced, now exit and make room for your kids. And that's all programmed into the genes of the different organisms. So, for example, mice, rodents might live 2 years on average. That's their program. And bowhead whales might live 200, and we're somewhere in between. But the aging reversal has been demonstrated about a dozen different ways in animals and in human cells. So, for example, we've taken cells from my arm and established and reversed their behavior, their physiology, to the point where they behave like embryonic cells. So you can take 80-year-old cells and they will behave like 0, and then you can reverse it so that you can differentiate them into any cell type you want. You can turn them into liver cells or brain cells or whatever have you. So you can turn that clock back and forth multiple times. We're getting quite good at that. Doing it at the whole organism level, where every aspect of your age is reversed, that it's not clear we have a good roadmap for that. But certainly, we've shown with a gene therapy that we can reverse at least 5, maybe 8 different diseases of aging that have nothing in common other than that they're age related. And we've done that in mice, and now we're doing it in dogs. And
so you may not be able to fix one part of the body, but you may be able to make someone's face look taut again and cosmetically nice, that kind of thing. One thing or the other.
It's more likely you'll fix the health related things before you fix the cosmetic things. But it's hard to predict which ones will be easy and which ones will be economically motivating.
Do you think if this happens, this is would be a therapy that would be available to many, or would it be a very expensive therapy? More wealthy people would probably be able to start with it first. I mean, there'd be a lot of ethical implications. Maybe, I'm not sure. But
part of the attraction of this for me is that it could be less expensive. So we've brought down the cost of reading your genome by 20 million fold. We'd like to do a similar thing for therapeutics. And since there's a kind of a fixed cost of doing the research and doing the clinical trials where you test the placebo and so forth on a large number of people, if those fixed costs can be amortized as paid off by a larger set of people, so if you have a very rare disease and you have these fixed costs of say $100 million, and you only have 100 people to benefit, then they're going to be paying $1 million each. But if you save $100 million and you apply it to 1 billion people, now they're going to be paying less than $1 each. And so it really matters how many people are affected. And in this case, aging affects almost everybody. I mean, there are few people that die young, but here in 2021, there are a lot, maybe 90% of the people in industrialized nations are dying from diseases of aging. So that could be a really low cost because you're spreading it out over a very large number of people who can benefit.
And it seems like when I listen to you and research some of the things you're studying, you have a proclivity to lowering costs. You want to expand and open these technologies as many as possible. And I think that's a good thing. And if you're going to be well known for something that, or famous for something that you do, which you seem to be, you might as well make a positive and contributing, impact to all. I want to keep things moving a little bit because I know your time is probably a little bit limited. You've been involved with co-founding 22 companies. Which one company are you the most passionate about? Something that you want to mention and something that you think is exciting to you?
Well, actually, I've co-founded 38.
Oh, sorry.
No, it's understandable. It's a fast-moving field. it's hard to pick one that stands out. I mean, certainly one that could buy us some extra years. So the one we've been talking about, Rejuvenate Bio, is a company that is aimed at buying us a few more years after we spend 66 years programming my brain, but education. It seems like a pity to just shut it off. So that's a kind of like, which one wish would you ask for from the genie? Well, more wishes. So that's how I can pick that one. But some of the other ones are very basic enabling technologies, and therefore the reason I'm excited about them is they can affect many other fields. So, for example, next generation sequencing, which we played a big role in, can be applied to everything from crime scene investigations to archaeology to medical and agricultural. You really can't write DNA without being able to read it. So everything that we've been talking about in terms of synthetic biology and therapies depends on that ability to read. And so that bringing that ability to read our genomes into existence is probably some of my favorite companies.
So the work and what you're working on, you're really a dissemination of information that's spreading out into other fields. So you're really not just contributing to gene technology or gene related businesses. Your work is flowing outward into many different types of businesses and abilities to learn and abilities to prove culture. And that's what you're saying in a sense, correct? A little bit.
It's nice to be exposed to all these different fields. It's intellectually interesting, but it's also you feel like you're having an impact on more than just a tiny field. And it's just nice to see a lot of people, whether they know it or not, using your inventions.
Well, I just want to say that, having the ability to talk to you today, I'm definitely having a very positive impact on me. And I know it's going to have a positive impact on a lot of people that listen to this. I want to just talk a little bit about, or maybe you could give us a little understanding of epigenetics and how epigenetics is affecting or enhancing your research.
Well, one form of epigenetics we've already talked about, which is our ability to take how old the cell thinks it is. So epigenetics is how you start with the genetics of the DNA and turn it into everything that it produces, all the proteins and the shapes. That's the epigenetics. And so aging is an epigenetic program by which you start as an egg and you develop into a baby, and then a reproductive adult, and then finally age and die. That is epigenetics. And each of the cell types, your eye cells and your liver cells and your hair and your brain, those are all epigenetics. And we've gotten very good control over it now. It's not just a kind of a weird phenomenon. It's something that a number of companies are based on. So, for example, GC Therapeutics, one of my recent startups, has evidence that they can make any stem cell into almost any other kind of cell in your body, and maybe multiple cells simultaneously to make organs for transplants or for testing new drugs and so forth. So I think we're in better and better control of epigenetics, and epigenetics can impact almost every disease.
But epigenetics, is that you changing the gene or having the ability to identify and change it into what you want to, or there's an environmental influence? I thought there was some environmental influence with epigenetics, and whether you smoke, whether you drink, your emotional state, or things like that, and how your genes are affected by outside external factors. Is that incorrect?
Oh no, that's correct. So you can change your epigenetics genetically. So you can put in an epigenetic regulatory protein.
Right. Okay.
Genetically, you can affect epigenetics by environment as well. So you can, if you expose somebody to a lot of ultraviolet radiation, then you're going to change the way that your skin program plays out. You can get, darker skin, more wrinkles, more skin cancer. All of those are epigenetics. You get the same genome, but you're changing the way that it plays out.
I had a question. I wanted to see external environmental factors on genes might affect when a child is conceived. I'll give you an interesting example, and I don't know if I'm way off the mark. When our son was conceived, we were both in really good shape. We were, running long distances. We were working out. We're both athletes, and my son was born, and he's physically gifted. And I'm wondering, is that pure? Do you think that would be a situation where it's pure genes, or maybe how we're influencing our own bodies and how we're taking care of it and how we're working out, and the fact that we were in both incredible shape at the time? Is that a sort of sci-fi question, or could there be some reality behind that,
Well,
George?
it sounds like it's spoken as a proud parent, for one thing, as you
Thank
should
you.
be. I think you can think of it on the flip side. If you were both abusing your bodies, in other words, taking serious addictive drugs, not caring about what you ate, or maybe possibly starving, either of your own work or just because of circumstances, that would result in probably health damaged children.
Mhm.
So the fact that you're doing everything right is the opposite of that. So it has a big impact. But it also heavily depends on genetics. You can change
Right.
one base pair, one ACG or T out of 6 billion in the inheritance of your child, and that child can go from being very healthy to extraordinarily sick, and often dying at birth or a couple of years later, with a lot of pain and suffering for the whole family. So it is both environment and genetics, and you need to get it all right. Fortunately, a lot of us do get it all right.
Right, for sure. I appreciate that description. Do you think that autism is I mean, obviously, there's genetics play a role in autism, and autism is prevalent today. I have a number of friends whose children are autistic. Do you think that there's some environmental mixed in with that, or it's purely genetics between the two individuals?
There's almost everything that we experience is a combination of environments and genetic. Autism. So autism is known to have a big genetic influence, but it also is influenced by the environment in ways that neither the genetics nor the environmental influences are fully understood for autism. But
Yeah. So I guess what I'm saying is that environment can turn on and off some of that sequencing, in the 23 pairs of chromosomes that are promoted by the genes, if I'm saying this correctly. Environment can sometimes switch some of those things on and off.
Sometimes it can make a lot of change, and sometimes very little. It depends on the underlying genetic problems and the kind of environmental change you're making. An example of something which you can think of as either 100% genetic or 100% environment is phenylketonuria. This is the sort of thing that you'll see on beverage cans that say this has aspartame. So
Aspartame.
don't take it if you have PKU. Well, if you know the genetics, 100 years ago, it was 100% genetics, meaning if you had this particular PKU inheritance from both parents, so you were doubly damaged, you would be severely mentally disabled. Very high probability, like 99%. On the other hand, if you fast forward to today and you have the right diet from birth because you know you're at risk, then you're completely intellectually normal. Again, very high probability, 99%. So you can turn something that's nearly 100% genetic problem into something that's nearly 100% environmentally predictable. So
through education as well, is what you're saying.
You're
And the
educating
knowledge
the parents. You take almost every baby gets a heel prick of a little bit of droplet of blood taken, and they diagnose 40 different genetic diseases that way, and all of which are actionable, that you can change something about the environment of the child and have a completely different health outcome for that child, rather than dying young and painfully. You live on, have a rich, full, healthy life.
Yeah, it's great. I mean, I've been watching my 23andMe account, and they've been slowly, updating with more information about your health and your traits and your genome and your genes. And so it's really interesting to see that. So you could actually, in addition to getting pricked on the finger, you can now self-educate yourself. And 23andMe most likely wouldn't exist if not for the work that you've contributed. That's got to be a truth, right? These companies would not be able to do if you didn't make understanding the genes and mapping the genes accessible. Don't you think?
It's been a team effort. There've been a lot of my close friends and colleagues who we have collectively produced that kind of technology. I did work with the founders of 23andMe as they were just getting started. I think most of the technology they used was developed at companies like Affymetrix and Illumina. So yeah, it's a wonderful team effort. Around the world, people are contributing ideas, and companies rise up and make them work and deploy them into the public. So it's that's
Very
true.
cool.
But, you know.
Very cool. Well, I kind of like put those two together. I didn't plan on talking about it. I know we're sort of moving along here with time. I wanted to just ask you one or two more questions. And I know you're a computer programmer and you understand zeros and ones and silicon and semiconductors. You talk about Moore's law. Moore's law is related to the exponential growth, or the how many transistors basically after a given amount of time you can put on a silicon wafer and increase processing power, and you compare that to synthetic biology or just biology in general or gene therapy, but you're saying that biology is moving much faster than Moore's law. Is that correct?
Yeah, I mean, both of them probably have limits, but biology, molecular biology and biotechnology, and particularly reading and writing DNA, have documented cost curves and quality curves where they've improved 20, 30 million fold, most of it in the last decade. And this has ramifications for everything that they touch. And it's not too surprising because, out in the field, at the forest and the agricultural environments, you basically get almost free production of many millions of square kilometers that are essentially just using atoms from the dirt and photons from the sun to produce these acres of atomically precise complexity.
Wow. All right. Well, you're getting a little bit beyond me today. You're definitely complex, but I understand. But what I think, no, it's not a bad thing. I was just trying to process your last few comments. But I think what I'm saying is, and what you're saying is that biology is going to progress very, very quickly, or maybe even a little bit more quickly than silicon technology, in a sense.
It has progressed more rapidly, say, since about 2004. It doesn't need to progress that rapidly to have a huge impact. Both computing, all aspects of computing, the chips, the disk drives, the optical fibers, they're all improving exponentially, as is biology. And it doesn't really matter whether it doubles every year or every 2 years or every half a year. It's still going at a breathtaking rate, and we all benefit from it, especially if we're thoughtful about safety and security and making sure there's oversight.
One of the other things that just reminds me about when I was reading about you was that you were talking about storage in DNA, and I couldn't understand. Did you mean actually storing data in DNA?
Right. So this was a kind of a wacky idea back in 2011 or so, or maybe even 1986. An artist named Joe Davis was thinking similar thoughts. But now there's a international consortium of companies that is storing data this way, and movies have been, and audio and all the digital things in our life have been stored in DNA now. It's about 1 million times more compact than the typical disk drive. It lasts a lot longer, so the record is about 1 million years. So it has good longevity. It doesn't. It has the same format that it did 1 billion years ago, at least in terms of its structure. So it has a few advantages. It's easy to make copies. We made 70 billion copies of my book as one of the first demonstrations of that. So it definitely has advantages. We've stored a trillion bits of information in a mouse, recording some aspects of its developmental biology and physiology in just 1 billionth of the mass of its body. So you don't have this big disk drive inside the mouse. It's just scattered throughout all of its cells.
Really? And you can read it back through all its cells too?
We have used that method to learn quite a bit about the individual experience of the development from egg to adult in many different mice. So we've learned things about that process. It's kind of like make the analogy with the black boxes in the airplanes that are constantly recording all kinds of data about the flight. And then you can choose to not read it at all, or if nothing happened, or you can read selected parts of it to find out, to try to debug what went wrong. So hopefully that kind of recording device, it might be something that
how do you pull it out?
Will find its way into medicine.
Yeah. How do you pull out the information?
You go to whatever cells you're interested in. So for example, we compared the left and the right brain to see when they developed, rather than the hind brain and the forebrain. And so you just pluck out a few cells, and then you can determine what their lineage was, from the single cell divided into 2, into 4, into 8, and so forth, all the way to the brain. And you can sort that out just by plucking a few cells and using the sequencing method we were talking about earlier.
All right. Well, I have to say that is amazing. That is really incredible. I might be wrongly assuming, but I think that technology might help scientists in understanding how the brain works, the functioning, I'm not sure. It's just a hypothetical question. The last question, and that was truly amazing. Thanks, George. The last question I want to ask you, and whenever you're speaking to a great mind, you always want to come up with one last big question. And my last big question for George Church is, and by the way, I've really enjoyed our conversation today. Where do you see humanity evolving to in the next 100 years? If you had to look out 100 years, and I know that you're smart enough, and I know that you look out that far, what do you see?
Well, so 100 years is not that far away. It's like 10 cycles of 10-year clinical trials. But it's a lot more can happen in the next 100 years than happened in the last 100 years, because we're going fast. The technology is going faster, and we have better observational and engineering capabilities than we've ever had. I think that one of the things that could happen is that with the improved healthcare costs, we could eliminate a lot of the diseases of poverty and aging, and that would help us solve other problems. Once we're not spending so much of our resources and our time on psychiatric disorders and diseases of poverty, then everybody can have a chance at a better education. Better education worldwide will result in another positive round of influence, more health. And once we get to the point where we really have this virtuous cycle of improving health and education, then we might have enough wealth to start thinking about our long-term survival. We're the only species on the planet that really has the ability of protecting ourselves and other species by making backup copies in other planets or even further out in space, because we're sitting ducks the way we are now. Asteroids, solar flares, all sorts of things that could, if we put all our eggs in a very small basket. So I just hope that within 100 years we will have solved enough diseases and poverty that we will feel that we have the luxury to be able to protect ourselves from these long-term risks.
And before we get annihilated by the solar flare, right?
Exactly.
So you're probably a fan of the interplanetary travel and what, SpaceX is doing. You're probably a fan of SpaceX.
Well, I've been a fan
NASA.
of NASA since I was a kid. I grew up in Florida, and so Cape Canaveral was a source of great pride. But I think the emphasis, since the 1960s and still to this day, has been on the physics and chemistry of getting us launched, and not enough emphasis has been on the biology of how do you actually make a self-sustaining colony. There's never been a truly self-sustaining colony that's completely isolated. The International Space Station, submarines, there's a lot of flux to and fro, and that might become impractical if half of the human population is not on Earth. So I think we should test that on Earth. We should make lots of little colonies on Earth, let them kind of compete economically, and work out the bugs where the consequences are not high. So if something goes seriously wrong with your colony on Earth, you kind of open up the doors and you go to the nearest emergency room. But if you're on Mars and something goes wrong with your colony, then the colony dies, and because it's a vacuum and it's cold. There's really nowhere to go. Anyway, I think the time has come. We wouldn't want to put it as a priority above, curing diseases of poverty, but it is time that we can do it in a way that is sort of equal cost, where the cost of living in a colony is comparable to the cost of owning a regular home. If we can arrange that, then we can get a 2-for-1. We can get experience with running a space-like environment without the risks. And we need to do this pretty soon if we're going to be launching rockets.
So simulate these economies, these different colonies first before we go out there, is what you're saying. Testing,
let the Darwinian and capitalism, various competitive forces, debug. It's the trial and error I talked about earlier. Engineering is not a purely theoretical exercise. We need to have lots of people testing it out.
Figure out the chaos first. Figure out the chaos. Figure out what to do when the one guy in the colony wants to run the whole place. What do we do with that guy? And it's a very small room. But George, if I was sitting there or I was up wherever you are right now, you must be up in Boston. Are you around the Harvard area?
Yes, right.
Is that where you're doing? I think if I was sitting in the room and you had the proclivity and the time, you would probably continue to talk with me, and I would want to talk to you for hours because I find you fascinating. I find you interesting, and it was a real, it's not often you get to talk to people such as yourself. I talked to a lot of different people in the podcast, but I knew I was going to be very interesting. I wasn't sure I was going to go, but it was awesome. It was exciting, and I thank you for your time, and I thank you for joining the show, and I hope we have an opportunity to speak again. You're super interesting, and you're a really nice guy. Thank you so much.
Thank you. You're too kind, and I look forward to our next conversation.
Thank you, George.